you are watching Planet Physiology. Today I will be discussing MCQs on the topic muscle physiology. Okay, let's begin with the neuromuscular junction. The first question is, which is the first step in neuromuscular transmission after arrival of the impulse at nerve terminal? Options are A. Exocytosis of neurotransmitter, B. Development of end plate potential, C. Movement of synaptic vesicles towards the membrane, and D. Opening of voltage gated calcium channels. Okay, simple straightforward question. Yes, the correct answer is option D, opening of voltage gated calcium channels. This calcium influx in the presynaptic terminal initiates exocytosis of vesicles by stimulating their transport towards the release site. Next question. This is from MGR University question paper. End plate potential is characterized by A. Propagation, B. All or none law, C. Depolarization, and D. Hyperpolarization. Okay. End plate potential is graded potential that develops at the end plate region of the muscle, that is at neuromuscular junction. So it is a graded potential and is always depolarizing in nature, and hence the correct answer is option C, depolarization. When it reaches to threshold, action potential is generated within the muscle. Next question. Usual amplitude of miniature end plate potential is A. 0.5 millivolt, B. 12 millivolt, C. 30 to 40 millivolts, and D. 120 to 130 millivolts. Miniature end plate potential occurs spontaneously when nerve is at rest, and this is due to quantal release of acetylcholine from single vesicle. So the correct answer for this question is option A, 0.5 millivolts. Amplitude can vary between 0.1 and 4 millivolts, and it varies directly with the calcium concentration and inversely with the magnesium concentration at the end plate. So even though this was a simple question, just 1/3 of my students could answer it correctly. Okay, now here is a case-based MCQ. A 35-year-old female reported the physician weakness in muscles and tiredness. She also stated that she is fine when she gets up in the morning, but weakness begins soon after she becomes active and it improves again on rest. Cholinesterase inhibitor treatment immediately improved her muscle strength. What could be the diagnosis in this case? A. Lambert-Eaton syndrome, B. Myasthenia gravis, C. Parkinson's disease, and D. Muscular dystrophy. How to approach such questions? So even though question appears bigger, no need to panic. Read the entire question along with the options and then read the question again. Find the hints provided in the case. For example, in this case, the important clues are: first is weakness starts after activity and improves on rest. Second, cholinesterase inhibitors improve the strength. So these are the guiding points to get the correct answer, which is option B, myasthenia gravis. In case of Lambert-Eaton syndrome, antibodies are formed against calcium channels in the nerve terminal, and hence neurotransmitter release is decreased. It affects proximal muscles, and muscles respond better on repeated stimulation. So this is the main difference between myasthenia gravis and Lambert-Eaton syndrome. Okay, next question. Myasthenia gravis is characterized by A. Formation of circulating antibodies to the nicotinic acetylcholine receptors, B. Sustained contraction of the muscles, C. Most commonly affected muscles are those of limbs, and D. Formation of antibodies against calcium channels. I think now everyone knows the answer. It's option A, formation of circulating antibodies to the nicotinic acetylcholine receptors. There are two forms of this myasthenia gravis. In the first form, extraocular muscles are primarily affected, and in second form, there is generalized muscle weakness of skeletal muscles. In severe cases, all the muscles including diaphragm can become weak, resulting into respiratory failure and death. Next question. This is from MGR University. Which of the following acts postsynaptically, blocking the nicotinic acetylcholine receptors and preventing the excitation of muscle cell membrane? A. Botulinum toxin, B. Curer, C. Neostigmin, and D. Tetrodotoxin. Correct answer over here is option B. Curer. Let us see action of each. Botulinum toxin prevents release of acetylcholine, so it acts presynaptically. Curer is arrow poison and causes competitive inhibition of acetylcholine receptors on the end plate, so it is postsynaptically acting agent. Neostigmin is anticholinesterase, so it prevents action of cholinesterase, and tetrodotoxin block voltage gated sodium channels. So correct option is B. Curer. Now another case-based question. A 47-year-old female was admitted to the hospital for nausea and vomiting for 2 days and then developed severe muscle weakness and neurological symptoms including ptosis and dysphagia. She indicated that she had eaten in a restaurant the evening before the symptoms began. Lab tests were positive for Clostridium botulinum. What is its mechanism of action? A. It blocks reuptake of neurotransmitter into presynaptic terminal. B. It exerts the effects by acting centrally rather than peripherally. C. It prevents release of neurotransmitter from the presynaptic terminals. And D. It reaches the soma of motor neurons and prevents impulse generation. Okay, lab test is positive for Clostridium botulinum and you are expected to answer its mechanism of action. So just now we have seen botulinum is a natural toxin causing paralysis and various forms of it act by different mechanisms, but in general they all prevent release of neurotransmitter at neuromuscular junction. Hence the option C is correct answer. Now let us start with the questions related to structure of muscle. Sarcotubular system, the statements are A. Exists in kidney. B. Comprises proximal and distal convoluted tubule. C. Made up of T tubules and sarcoplasmic reticulum. And D. Comprises smooth and rough endoplasmic reticulum. Okay, a simple question, straightforward question. It is made up of T tubules and sarcoplasmic reticulum. Next question. Functions of transverse tubule in skeletal muscle is A. Rapid transmission of impulse to all the myofibrils. B. Storage and release of calcium ions for process of contraction. C. Pumping of calcium ions from sarcoplasm. And D. To provide site for nerve invagination. Again a simple question. Yes, transverse tubule is concerned with rapid transmission of impulses to all the myofibrils. Next question. True about skeletal muscle contractile response is A. Contraction precedes the action potential. B. Decrease in calcium concentration within the cell to 2 into 10 power minus 4 moles per liter produces muscle relaxation. C. Both contraction and relaxation of the muscle are active processes. And D. Calcium ions passively reaccumulate into terminal cisternae during muscular relaxation. Okay, the correct answer over here is option C. Both contraction and relaxation of the muscle are active processes. Now, if you see the responses of my students, it suggests that they do not pay proper attention to this topic. Remember that action potential always precedes the contraction. That is, action potential in the muscle leads to their contraction. So, option A is false. For muscle relaxation, calcium concentration should be less than 10 power minus 7 while at the concentration of 2 into 10 power minus 4 muscle contracts. Hence, option B is also wrong. Once action potential is over, calcium are actively pumped back into the cisternae, which is primary active transport process and not a passive reaccumulation, and hence the statement D is also wrong. Next question again from MGR University. Staircase phenomenon is due to A. Increased availability of intracellular calcium, B. Synthesis of troponin C molecules, C. Summation, and D. Tetanus. Okay, so this record shows the staircase phenomenon recorded in frog's muscle. The staircase phenomenon is progressive increase in force of first few contractions if muscle is stimulated repeatedly. It occurs due to increase in intracellular calcium concentration. So option A is correct answer. Another reason for staircase phenomenon is also heat generated by the previous contractions help in increasing the force of contraction. Another MCQ from MGR question paper. Which of the following muscles cannot be tetanized? A. Skeletal muscle, B. Smooth muscle, C. Cardiac muscle, D. All of the above. Okay, a simple question. Cardiac muscles cannot be tetanized, and the reason for this is their long refractory period. Next question again from MGR University. Fine irregular contractions of individual fibers are called as A. Fasciculations, B. Fibrillation, C. Spasm, and D. Twitch. Yes, the correct answer is option B. Fibrillation. Because question says these are the fine irregular contractions of individual fibers. Let us see the meaning of other terms as well. Fasciculations are visible contractions of group of muscle fibers that occur as a result of pathological discharge of spinal motor neurons. Spasm is an abrupt, violent, involuntary contraction of the muscle or the group of muscles. When a single action potential causes brief contraction followed by relaxation of the muscle, this response is known as muscle twitch. Next question. Troponin-tropomyosin complex is believed to play what role in muscle contractile process? A. It provides major amount of elastic tension during the contractile process. B. It is believed that in resting state, it covers the active sites on the actin filament. C. Combination of this complex with myosin excites the activity of power stroke. And D. Combination of potassium with troponin portion of this complex is believed to trigger muscle contraction. Okay, here is a diagram showing troponin-tropomyosin complex, and as you can note, at resting state, this complex covers the active sites on the actin filament. So option B is correct answer. During excitation contraction coupling, when calcium ions bind to troponin C, this complex moves laterally to uncover the active sites and initiate muscle contraction. Next question. The length of the muscle at which it develops maximum active tension is called A. Optimum length, B. Resting length, C. Equilibrium length, D. Initial length. Okay, correct answer for this question is option A. Optimum length. The length of the muscle at which muscle fibers can generate maximum force isometrically is called optimum length. The length of the muscle at rest in the body is its resting length. And usually in the body, resting length of the muscle is optimum length, where maximum active tension is generated. The length of the muscle before contraction begins is termed as initial length. So here is a diagram showing the resting length where active tension developed in the muscle is maximum. Next question. Tension developed in the muscle due to activation of contractile component is called A. Passive tension, B. Active tension, C. Total tension, and D. Resting tension. Okay, let us study this diagram again. Passive tension is the tension developed in an unstimulated muscle. And as indicated in this part, passive tension increases as muscle is stretched passively. When muscle is stimulated, we record total tension. This particular graph indicates total tension. The difference between total tension and the passive tension is the amount of tension actually generated by the contractile process and is called as active tension. Hence, the option B, active tension, is the correct answer. Next question. The muscles typically exhibiting isometric contractions are A. Extraocular muscles, B. Respiratory muscles, C. Anti-gravity muscles, and D. Masticatory muscles. So, correct answer is option C, anti-gravity muscles. So, even though this is a simple MCQ, almost 50% of the students chose wrong option. Anti-gravity muscles maintain posture and support body against the gravity. And during isometric contraction, muscle length remains same. What changes is only the tension generated within them. Okay, coming to the next question. Red muscle fibers have A. High oxidative capacity, B. High glycolytic capacity, C. Fast contraction, D. Fast myosin kinase activity. And the correct answer over here is option A, high oxidative capacity. Red muscles are also called as type 1 fibers or slow fibers. And this is because they contract and relax slowly due to slower myosin kinase activity, and they also have less glycolytic capacity. If you want to revise this topic, link is shared in the description box below. Next question. Which muscle protein is affected in Duchenne muscular dystrophy? A. Desmin, B. Dystrophin, C. Myomasin, and D. Titin. Yes, correct answer is option B, dystrophin. All these are actually the structural proteins in the muscle. Dystrophin connects thin filaments to sarcolemmal proteins. Titin connects Z line to M line. Desmin anchors Z lines to sarcolemma, and myomasin forms M line and anchors thick filaments to titin. Here is a picture showing all these structural proteins. Now coming to the smooth muscles. Multi-unit smooth muscle is found in, this is the question from MGR University. Options are A. Uterus, B. Ureter, C. Intestine, and D. Iris of the eye. Yes, it's option D, iris of the eye. Another example of multi-unit smooth muscle is piloerector muscles. Next question again from MGR University. The property of smooth muscle of hollow organs to return to original force of contraction after elongation or shortening is. Options are A. Latch mechanism, B. Slow cycling, C. Stress relaxation, and D. Power stroke. Okay, here correct answer is option C. Stress relaxation. Stress relaxation allows hollow organs to maintain almost same pressure within them despite long-term large changes in the volume. For example, maintenance of blood pressure during changes in blood volume or pressure changes within the bladder. And the last question of the session. Latch bridge mechanism in smooth muscle is responsible for A. Fast muscle twitch, B. Sustained muscle contraction, C. Excitation contraction coupling, and D. Unstable membrane potential. In case of latch mechanism, after the smooth muscles develop full contraction, enzymes are inhibited and hence the cross-bridge cycling rate decreases. This allows smooth muscles to maintain prolonged tonic contractions for longer time with little use of energy. So the correct answer here is option B, sustained muscle contraction. Unstable membrane potential in smooth muscle is called as basic electrical rhythm or slow waves. So that's all for this session. Thank you for watching and see you in the next video. If you enjoy my presentations, press the like button and share it with your friends. For more such videos, subscribe my channel and click the bell icon. Thank you for watching and see you in the next video.